But allow me first to a small introduction to the company. ALK-Abelló is a pharmaceutical company working with allergy for more than 80 years. Right now we're approximately 1,400 employees worldwide, and the company has a turnover of about 200 million euros. We have a global market share of about 1/3, and we consider ourselves the world leader in specific allergy management due to our history and our global presence. We have production sites in Denmark, Spain, France, and United States. Also in United States, we produce raw materials, pollens in the state of Washington, and Hymenoptera venoms in Pennsylvania. We have research in Denmark, Spain, and France, and recently we opened a research facility in China. To give you an impression of our scientific activities, I've just shown some of our recent publications on this slide. Last year, in the year of 2006, 32 peer-reviewed articles in international journals were published with co-authors from ALK-Abelló. 6 of them concerning the new GRAS tablet, 20 of them concerned with characterization of allergens and immune responses of allergic patients. Shown on this slide are 6 clinical studies with Allotar SQ. The first one is the world's largest study of subcutaneous immunotherapy with more than 400 patients. And the first author is Anthony Frue, the president of the European Academy of Allergy and Clinical Immunology. Another publication from last year was the 5-year follow-up data on the preventive allergy treatment study, the PAT study, which was also mentioned by Professor Spitzak. Also, the first immunotherapy study in China with standardized extract was published last year. And a study showing effect on atopic dermatitis of house dust mite immunotherapy as well. 2 other studies described reduction in steroid use in asthma patients and improved safety of bee immunotherapy using alum formulation. But the number of publications may not reflect the true impact of this research. A better measure might be the number of citations, and shown on this graph is that last year in 2006, more than 900 papers cited publications from ALK-Abelló. We have a history of 30 years of allergen research and clinical research. In the 70s, the first allergen standardization program was the Danish Allergen Standardization Program in 1976, with the participation of ALK-Abelló for the development of scientific methods for standardization to control the natural variation in the raw material of for the production of products for specific immunotherapy. In the 80s, we were working with purification of single allergens, identifying the important allergens and purifying them for the purpose of raising specific reagents in rabbits for the measurement of major allergens in the crude extracts. In the 90s, the recombinant allergens appeared, and the research group at ALK-Abelló was the first to publish a paper on the characterization of recombinant allergens. We were also the first group to describe in a publication the 3-dimensional structure of an important inhalation allergen. This is the birch pollen allergen, BEDV1, and that was published in Nature Structural Biology in 1996. Also an interesting publication is the description of an allergen antibody complex, so that you can really see the atomic interactions between the allergen and the antibody in this crystal structure. The products of ALK-Abelló are primarily concerned with immunotherapy. The Allotar SQ for subcutaneous immunotherapy constitutes about half of our turnover. We also have the Pangramine SLIT products, which constitutes about 1/4 of our turnover. And the last quarter of the turnover are miscellaneous products, products for skin prick testing, and also the EpiPen, the emergency treatment for allergic shock. The new product, the GRAS tablet, has not yet any sales figures. So what's in for the future? Well, we have just successfully registered the GRAS allergen tablet in 27 European countries, and we are now negotiating prices and reimbursement in each of these countries. The tablet is on the market in Germany, United Kingdom, and Scandinavia at the moment. The next tablet to follow is a house dust mite tablet, which is now in clinical development. And then we have planned tablets also for ragweed and birch pollen allergy. Second generation tablets may be recombinant tablets containing recombinant allergens. We have a strong position in recombinant allergens with a portfolio of IP rights in the most important areas. And we have been active in publishing results from recombinant allergens for the last 15 years at a rate of approximately 5 publications per year. I think the most important aspect of the products from ALK-Abelló is the level of standardization. Standardization is necessary, as I said, to control the natural variability of the raw material. With a well-controlled allergen extract that has the same potency from batch to batch, we can increase the dose without having the risk of side effects. And we know that the effect of the treatment is dose dependent. So the higher the dose, the better the efficacy. However, the dose is limited by the risk of introducing side effects. So if we can control the extract, we can go to a high dose. If we cannot control the extract with standardization, we will need to lower the dose in order to make sure that we don't induce too many side effects. So a highly standardized product is more efficacious compared to a product with a lower level of standardization. And why is standardization an issue? Well, it is because of the complexity of the allergens to which the allergic individuals are exposed, the complexity of the individual immune responses, and the complexity of the materials we use for the treatment products. I'll just show you one example. This is a system called crossed immunoelectrophoresis, in which each of these blue precipitates represent one antigen in an allergen extract. And if we probe this plate with patient serum, IgE from the patient serum will bind specifically to the precipitate in question, and we can then stain with a radioactive anti-IgE and develop the picture on an X-ray film. So by this method, we can identify for each allergic individual which allergens does this particular individual react to. And as indicated here, there's a large difference between individual patients. If we perform this experiment with a panel of sera from allergic patients and we count for each precipitate how many patients react to this particular allergen, we can make a so-called allergogram showing the prevalence of reactivity to this particular component in a patient panel. And we will then get a typical picture like this. We will see that there are some of the components that are most often reactive in all patients. That's the major allergens. And then we have a large number of components to which we only rarely see a reactivity. So this is a way of identifying the major allergens for one. For the second observation is that we need to contain most of the components in the extract in order to make sure that it's it's efficient in immunotherapy. Now, standardization is more than batch-to-batch standardization. The standardization procedure at ALK-Abelló also is dealing with the control of the raw material, the process for the preparation of the extract, characterization of the extract with scientifically based methods, and as also Professor Spitzak mentioned, the importance of the in-house reference, the biological testing of the in-house reference preparation, and for most SQ extracts, this in-house reference has also been used in specific immunotherapy and proven efficacious in clinical trials. Of course, then also batch-to-batch standardization and formulation of the vaccine is part of the standardization program. Batch-to-batch standardization of all our products for each and every batch is based on assessment of complexity in the immunoelectrophoretic system, the measurement of the major allergen by rocket immunoelectrophoresis, for example, and the measurement of the total potency, that is the total IgE binding of the extract in a RAST or a RAST-based method. So we can have a strong batch and we can dilute it to get the same strength, the same potency as the internal standard, or we can have a weak batch and we can then concentrate it, but only if the complexity and the major allergen content is the same as the internal in-house reference preparation will the batch be accepted. Otherwise, it will be discarded, or as we most often send it to the research department and they can play with it. Now, the SQ unit was developed historically as shown in this sketch. As standardization advanced with time and clinical experience accumulated, at one point when standardization was so advanced as to be reproducible of the extract, we defined the optimal clinical maintenance dose as 100,000 SQ units, and there was then initiated double-blind placebo-controlled trials for each of the important extracts: grass, birch, cat, dog, and house dust mite. And all of these double-blind placebo-controlled trials were followed up after 6 years after termination of treatment to see if the effect was still effective. These are the references. And the interesting thing is that if we look at the major allergen content in the optimal maintenance dose, we find that the major allergen content in each of the extract lies in a very narrow range, and this is also published in the WHO position paper on allergen immunotherapy from 1998. The major allergen constant is remarkably constant between 10 and 20 microgram. So this is the effective dose for immunotherapy. Alutard SQ is the golden standard for immunotherapy products. It's by far the most well-documented product on the market. We have more than 65 peer-reviewed international publications on Alutard SQ, describing 23 randomized controlled studies, documenting both early effect after 3 months of treatment, progressive effect, that is, you build better effect as the treatment progresses, persistent effect, as I just mentioned, after termination of treatment. We have also seen the PAT study showing preventive effect concerning both new sensitivities and the development of hay fever into asthma. And we have a lot of papers describing the immunological effects, the effect on the immune system. Only with Alutard SQ will you get the documentation for all these different levels of efficacy. Even though all of the manufacturers claim their products to be standardized, the term standardized is not standardized. So each manufacturer has own standards and standardization principles that differ. allergen extracts from different manufacturers differs in composition, and therefore they cannot be compared. There are no common units or measures, and regulatory requirements are not strict enough, according to my opinion. For example, there's no regulatory requirement for the determination of major allergen content in each and every batch. So therefore, my recommendation would be each product should prove useful in double-blind placebo-controlled clinical trials. You cannot just say that immunotherapy is documented. Each product has to show its own, in its own right, efficacy. And you should ask your agent for clinical documentation and what standardization procedure is in effect in the company. So if you need more information, you are welcome to send me an email or look at our website. Thank you very much.
Já bych teď poprosil asi o dotazy na pana doktora, aby mohl odejít. Jestli máte nějaké cílené otázky na produkty firmy ALK. My first question is it so difficult to describe in data sheets of every immunotherapy preparation from ALK to say what kind of epitopes are present at least, because I always wonder if I do prick tests and then immunotherapy that both contain the same epitopes so that I can at least guess that I use proper way of treatment.
It's the same extract. So you make a bulk extract, standardize it, and this extract is then distributed into the different products. Like it's formulated in aluminum hydroxide for Alutard. It's formulated for sublingual drops or for the prick test products. So it should be the same epitope.
It should be, but it's a question because it's different media that you use.
Yes.
It's for sure that three-dimensional composition is maybe a little bit different. I saw this year first time poster from your company in San Diego meeting where you tried to identify epitopes present in vaccines that were used for immunotherapy, and that's, according to what I know, first report that you tried to identify and confirm the presence of the epitopes.
Exactly. I mean, it has always been said that once you have formulated your extract in aluminum hydroxide, you cannot analyze it. But we are trying to do that anyway, and what we have found so far actually confirms that there is no dramatic change in conformation when the allergen binds to the aluminum hydroxide gel or when it is taken off again. So it seems that, but of course we cannot generalize. Each allergen has its own characteristics.
And what about recombinant antigens? Does your company intend to use it, and do you see advantage in recombinant antigens?
Well, of course, we have this long experience with the recombinant allergens, and personally, I have been working very much with them very early and would like to see them in the products. But when it comes down to the bone, there's actually no clinical advantage of the recombinant allergen. There's manufacturer advantages because you can avoid all the standardization problems and you can have a homogeneous, well-defined reagent, but that doesn't give you any clinical benefit. If you think in terms of modifying the recombinant allergen, you may have a concept for improved clinical benefit, but to use just the naturally occurring sequences in the recombinant form doesn't add any clinical benefit, and it will be a costly development program. Now, we have experience from the grass tablet clinical development program, which costed around 150 million euro, and if you cannot have any clinical benefit, you cannot justify such an investment. So I think that's the reason we are not currently conducting any development programs with recombinant allergens.
Máš ještě jednu otázku?
I have a question. You declared that indication for sublingual tablets house whites is rhinitis and asthma, and using Rhasis or Reglit is only rhinitis. So why is the difference in indication? I noticed that indication for Reglit tablets is asthma and rhinitis, and for Rhasis and Reglit it's only rhinitis.
I agree. I think it was an error in correction of the slides. I'm sorry. No, I don't see any reason for any difference there. I agree with you.
Ještě jednu otázku. My last question, when you compare from various points of standardization batches from, for example, year 2006 and 2005, they are always comparable or they are different?
Well, thank you for that question. Of course, we didn't do that analysis, but actually we did, because in Denmark, and I have a little bit red ears, because I have to admit that we had some problems with, suddenly a lot of adverse reactions showed up in Denmark with the Fleum pratense product, and everybody immediately blamed the product. So we, in the research department, we dropped everything we had in our hands, and we searched all the freezers in the company to find all the batches that we could get our fingers on, and we even developed new assays to look at the product in different way. And I'm happy to say that this effort actually showed that it was remarkable constant, and of course I don't have the documentation, so you will have to take my word for it. But the conclusion on the case from the Danish allergologists was that what was needed was education. So now they have education programs for GPs. A lot of the reactions were very quick reactions within 1 or 2 minutes. So the conclusion was that it was injection into the vein or the bloodstream. So we couldn't, there was no problem with the product, I'm happy to say that.
Is it so difficult to include in data sheets to every immunopreparation more of your company standards, not only to say that it was standardized according to your internal house rules, but to say some more details so that at least I can take 2 different papers and can compare, that you guarantee something, and if something happens, that it's my mistake or your mistake. And if you put some information on this paper, then you also have to think twice whether is everything correct, and you take the responsibility on what you describe here.
We can describe what is part of routine quality control, and we do that. But of course, these products are complex mixtures, and we don't know everything about every component in the extract, and also we don't know all the components that are not proteins. I guess if you had to register such a product today, it would probably not be possible. So I think we'll have to live with this as long as we use the extracts, that we can't explain everything.
Right. Thank you very much. Thank you. Ještě někdo máte otázky nějaký? Ne. Máte ještě někdo? Jo. Já bych teda ještě možná využil té možnosti, že bychom mohli položit otázky panu doktorovi Řehákovi, jestli máte někdo otázky na laboratorní diagnostiku. Existují, pane doktore, nějaké podobné standardy, které by teda byly někde dokumentovány ohledně alergenů, které jsou pak použity při té laboratorní diagnostice, které epitopy tam jsou přítomny, které nejsou, jestli to jde někde dohledávat, když mě to bude zajímat.
Tento dotaz jste mi už pokládal.
Jo.
A když jsem se ptal o firmy, tak jsem byl ujištěn, že veškerý bezpečnostní listy a tyhle věci můžete mít, ale tam to, že je zatím kryto tajemství obchodním, že to nevyzradějí. Nevím, jestli se situace změnila, můžu ten dotaz položit znovu.
Ne, já se spíš ptám jenom, protože když pak člověk použije několik těch kritérií, klinické kritérium, kožní kritérium, laboratorní kritérium, tak je to takové...
Zatím jsem byl odbyt, to nebylo jenom, když jsem to...
Děkuji. Ještě k panu docentovi Kučerovi, jestli máte otázky nějaké. Já jsem se ptal na pohlaví vzhledem k té diagnostice a ještě věk, tak si tam zmiňoval u dětí, že je něco jiného ještě než u dospělého člověka. Jak je to u lidí třeba věku 70 let a 40 let, je ta kožní reaktivita a diagnostika alergií taky stejná, nebo jsou tam nějaká rizika? Citlivosti a specificity.
Obecně, myslím si, že zrovna dánští pracovníci udělali velkou studii, která sledovala množství celého IgE v populaci zdravých jedinců, a to je zcela jasný pík mezi 12 a 25 lety a poměrně pozvolný, ale trvalý pokles, a to věku asi 70 let. Pokud to volně interpretovali, že třeba obecně zvýšené hodnoty opírují tuto křivku, tak ta klinická zkušenost to potvrzuje, že tyto diagnostické postupy, řekněme, ta detekce kožní testem IgE je spolehlivá už 4 let věku a trvá až do vyššího věku a stane se ta odezva nebo velikost kožního testu, ty hodnoty budou nižší v tom 5., 6., 7. věku.
Děkuji. Ještě máte někdo nějaké dotazy? Ještě?